All right, this is our introduction to molecular biology. We're going to discuss the molecules of inheritance and how they work in cells. So, this is considered the central dogma of biology. And it really, we now know the genetic code. We know how cells make traits, and that's by making proteins. So, I talked a little bit about this guy. Um, in the turn of the 20th century, we recognize Mendel had solved the basic patterns of inheritance, but there were questions: how did this work? What were Mendel's factors? They must be in cells. These inherited factors, or we now call them as genes, also came to be recognized as responsible for human genetic disease. So Garrod was the first one to connect this with proteins. That in many genetic diseases, that there was a protein that was missing or it was malformed. And he proposed that patients with these kind of diseases were missing an enzyme, and those were all proteins. And so it connected the trait from the gene to a protein. Then it was up to biologists to figure out what exactly was the gene and how did it make a protein. So these inherited factors were clearly associated with chromosomes. In chromosomes and mitosis and meiosis moved like we would expect the Mendel's genes to move. And in karyotypes, we recognize that we could see the chromosomes and see that in different species, the chromosomes were different, which is what we would expect. As we got better and better at things, we could transfer nucleuses and see that the nucleus determined the traits of the cell. And then in other experiments, we identified that it was the DNA in the chromosomes that actually inherited the traits. And so Rosalind Franklin's work was crucial in that. She was the one that had the most important evidence, and that led us to the structure of DNA. Okay, and that was the key factor. In the nucleus were the chromosomes, and in the chromosomes was DNA, and DNA was the genetic material. Okay, DNA is one of two types of nucleic acids. These are really large molecules. Deoxyribonucleic acid was mostly in the nucleus and clearly in the chromosome. RNA, ribonucleic acid, was mostly in the cytoplasm but had a very, very similar structure. These were both built of nucleotides. Okay, nucleotides were a component of both RNA and DNA. Each contained a sugar, a phosphate, and nitrogen bases. In DNA, the sugar was deoxyribose, and in RNA, it was ribose. There were different bases in DNA and RNA, slightly different bases, but the phosphates were exactly the same. And these were linked together by phosphodiester bonds between the sugars and the phosphate. There's a picture of a nucleotide. That green spot on the bottom, that's the only spot that differs in the sugars between DNA and RNA. In DNA, there's a hydrogen there. In RNA, there's a OH group. Then there are 4 different nitrogen-containing bases in DNA and in RNA with only slight differences. Okay. In DNA, you have adenine, guanine, cytosine, and thymine. Okay. In RNA, uracil replaces thymine. Otherwise, the other 3 bases are the same. Here's a single-sided nucleic acid chain showing the nitrogen bases exposed on one side and then the sugar-phosphate backbone linked together. Okay. DNA then was found in the chromosome and structured kind of like this as a double helix with 2 chains linked together by hydrogen bonds between nitrogen bases. Adenine was always across from a thymine and cytosine always across from the guanine. This sequence of 3 bases served as a code for making a protein. And that's where the information was. And once we knew DNA structure, it was easy to see that, okay, the code must be here. And it became really easy to see how the molecule could be copied. The 2 strands could be separated by just easing apart these hydrogen bonds, and then a new strands could be made that would match the old strand by just pairing nucleotides that had the right base. So this was a really key thing in understanding it. And we also found enzymes that could make an RNA copy from a DNA. And so we quickly realized that the central activity of virtually all cells was that DNA would make an RNA copy, and that RNA copy would be made into a protein. Okay, and we found that this process occurred in all cells. In all cells, it was a two-step process where one enzyme copied DNA into RNA, and a second set of enzymes copied RNA into DNA. And so this is what's called the central dogma. Originally found in E. coli, but quickly realized that the same process, the same code is used in all cells, prokaryote and eukaryotes. Okay. It was also quickly found that, a very easy process to copy DNA or DNA replication used many enzymes, but one basic enzyme called DNA polymerase. And then the two strands would be separated and new strands would be built using old strands as a template. And by just matching complementary nucleotides across. And this enzyme made very few mistakes and could even repair mistakes. So between these two things, the DNA replication and the central dogma of making protein from DNA, we came very quickly to recognize, okay, this is it. This is the central activity of all cells. Okay, and particularly this making of a protein, this two-step process. Transcription, making RNA from a DNA template. That's the enzyme that does that is RNA polymerase, shown right here. And then translation, the second step, which the main enzymatic element here is a ribosome. And this ribosome copies the RNA or translates the RNA into a DNA or a protein sequence. Okay, and so these things are, once we understood DNA structure from understanding the XY crystallography data, it was really easy to understand what was going on. And you could see, okay, here's the enzyme separating the DNA strands, and while it separates the strands, it builds a copy based on matching nucleotides. A matches T, T matches A, C matches G, and G matches C. Then this RNA sequence would go into the cytoplasm where a ribosome would assemble on it, and other enzymes would recognize these little codons, which are the 3-base sequence, and bring the correct amino acid. When they do that, the ribosome links the amino acids together so that they start forming a chain of protein. Okay. Sir Francis Crick was instrumental in doing that, and another guy in the movie, Sidney Brenner, also was really important. They determined that the DNA code and how it led to an amino acid chain. The code involved 3 nucleotides, and each of these nucleotides would be read as a group, and then that sequence could be changed into an amino acid. That also showed us how protein sequences could change over time, because changes in the DNA sequence would lead to changes in what proteins were put there. So it allowed us to recognize mutations and how evolution occurred. And based on lots of work, we were able to identify that the genetic code, here are the sequences of the RNA nucleotides, led to all 20 amino acids could be installed into a protein. And there were both, not only were there codons for amino acids, but there were start codons like the AUG here, shown in green, for methionine. Virtually all proteins start with that. And then there were specific stop sequences like UAA. So this probably took us, after Mendel, it probably took us like 70 years to do this. But we found the code was universal. It was the strongest evidence that suggested all living things share common ancestry. It led to advances in genetic engineering where we could insert genes from one organism into another. This pig on the left here has a fluorescence gene inserted into its genome, and it produces this fluorescence protein. And so this guy will glow green under ultraviolet light. They also led us to identify how mitochondria and chloroplasts work, and that they have their own gene sequences that are slightly different from ours. So we're going to explore this more. We'll go into more details on how these making proteins and copying DNA works, but we'll also talk about a lot about genetic engineering and the advances there.